Welcome, folks. We're going to wait just a minute to get started as more people are just starting to join. Here we go. Excellent. Well, I'm really delighted today to welcome Dr. Jonathan Kipnis. He is the Alan and Edith Wolf Distinguished Professor of Pathology and Immunology at the Washington University, St. Louis, and has been lighting it up for years, but especially this past year, regarding things like brain clearance, the brain and immune system interactions. And so we're going to get into that today, and it has lots of practical implications for our sleep, our health, and other matters. So welcome, Jonathan. It's great to have you.
Thank you so much. Thanks for having me. Super excited.
Well, I thought what we'd do is start with in the journal Neuron. You had the cover article with. I love the cover. It's a mop it's a mop in the shape of a brain.
Yes. Yeah.
And, basically, I kind of look at what has been developed over time is like our brain has a dishwasher that works at nighttime, and it's on taking out the trash. But maybe what we can do is start firstly that the brain. There was a misconception about the brain, its immune system, and these fluid dynamics for years. Maybe we can start with what was wrong and what's right now.
So, maybe I'll give a little bit of introduction, like what you're saying, right? So basically, every tissue in our body has two types of vessels. Even let's make it even simpler. Every household has two types of pipes. We have the water pipes and the sewage pipes. And so water pipes are bringing the water, sewage pipes are removing the dirt, right? And so if you want to clean something, you get fresh water, you clean, and you pour it into the drain. So funny enough, our tissues work in a very similar way. We have the blood vessels that bring the oxygen, the glucose, nutrients into the tissues. And then we have the lymphatic vessels, which people don't know maybe as much about lymphatic vessels. And those are the sewage pipes of the tissues. And so they remove the metabolites and the used, if you used water from the tissue out. And that's how the tissues are being washed. Now, the lymphatic system is a sewage pipe taking the material from the tissue, the dirt, the waste, into what is called the lymph nodes. And those are the headquarters of the immune system. And the immune cells are sitting there and just walking through the trash. Just imagine your neighbors are going through a trash can.
Mm-hmm.
They will know everything about you, right? Now, if something weird comes in your trash can, something like really concerning, they may call the police to understand what's happening, why there is, I don't know, whatever is in the trash can. Same thing with the immune system. When the immune system recognizes that something is abnormal, unusual coming from that tissue, they go into the organ to understand what's happening. For example, viral proteins, abnormal proteins, so bacterial proteins, some things that are not supposed to be in a tissue, immune system sees, and then they go and patrol. That's called immune surveillance. So the idea was that the brain doesn't need immune surveillance. So our most important organ in the body does not need immune surveillance, doesn't have it because it doesn't have lymphatic vessels. Now,
Was that ever wrong? Wow. Yeah.
Well, so it was funny enough. In 18th century, there was an Italian physician, Mascani, who claimed that there are lymphatic vessels on brain surface.
Mm.
And then he was proved completely wrong. He died in he died in complete infamousy. Nobody knows even where he's buried. And there was this very famous Lancet article. As you know, Lancet is like a top medical journal.
Sure.
That described how amazing Mascani was and what his 4 mistakes are. And mistake number 1 is lymphatic vessels on the brain surface. Okay, and so we were interested in my lab, to understand immune-brain interaction. And because there is very little immune cells inside the brain, but a lot of immune cells in the outside, in the membranes. Maybe it's a good point to introduce what meninges is. So between our brain and the skull, we have a layers of, if you wish, a cushion material, which we call meninges collectively. And so people may know disease meningitis, which is infection of meninges. And so this is the meninges, and meninges is very rich in immune cells.
Right.
The brain itself is very poor in immune cells.
Yeah.
And we were looking at meninges for different immune subtypes and all this. And then we stumbled upon something that looked like vessel. So we came to our colleague back in the University of Virginia. We came to our colleague and we said, do you have a good marker to label for lymphatic endothelial cells? And he said, why would you need it? Don't you work with the brain? There's no lymphatics in the brain. And we said, yes, but we think we see something, maybe. And they gave us the material, the antibody, and they said, here, waste our reagents. So we went back and we labeled, and voila, it was real. It was a vessel. So basically now we do have vessels. We do have these sewage pipes but they're not in the brain. They are outside the brain.
Well, you discovered meningeal lymphatics.
And we discovered those. Yes, exactly. Yeah.
That was back, I think, 2014 or something?
No, 15. Yeah, 10 years ago. Exactly 10 years ago.
Yeah, 10 years ago. But now let me try to reframe this because there is this process in the brain that is obviously relying on lymphatics, right? So
Yes.
that is preparing, getting that stuff into the sewage system, if you will, right?
Yes.
Um,
So it's a very complex system of fluid flow through the brain. And then, so again, in normal tissue, the fluid would come with the blood vessels, if you wish, and immediately would leave with lymphatic. So very, very easy perfusion, very easy washing. In the brain, we make a fluid. It's called cerebrospinal fluid. Right? We make about half liter of it a day, which is about 3 times the volume that we normally have. So we replace it 3 times a day. The fluid not only goes around the brain and the spinal cord, but it also gets inside. It goes in, fresh fluid goes in, washes through the brain, and then comes out and then leaves through the lymphatic vessels.
right, right, right. That's so now the clean cerebrospinal fluid, it's now in the brain, and this lymphatic system is being the channel for getting the waste products out. And surprisingly, waste products, metabolic waste products, but proteins like beta amyloid, right?
Yes, well, beta amyloid as well. So basically, anything that is produced in the tissue and needs to be washed out is being washed out. Some of it are maybe very bad metabolites. You want to get rid of them. Some of them are amyloid beta, which you don't get rid of them, they will become sticky and will stick and will accumulate in the brain. Some of them are proteins or even pieces of cells, which are not necessarily bad or necessarily good, but they are being taken out to then again be introduced to the immune system for surveillance, and that becomes very important because the immune system sees the picture of the tissue.
Right,
When
right,
tissue's health is changing, then immune system will see a very different type of picture.
right.
At which point they may decide to react to the problem that's happening in the brain.
Okay, so now before we get to the exit out of the brain,
Yes,
I want to talk about stuff that's modulating inside the brain, this
yes,
lymphatics, and it looks like there's some three. This is going from the neuron cell review, which is just in my view, a masterpiece to bring it all together up to the moment what we know. But there's three ways that you can modulate this lymphatic collection, and that is through the artery pulse, the
right,
respiration, working through the
right,
vacuum of the veins, and then what you call vasomotion. Uh,
right.
and there's a nice diagram we'll link with that paper and show the graphic. Um, so those are the ways that you could physically change the flow of the lymphatics. Is that right?
There is, yes, what you said is absolutely right. There is even more than that, right? So there is, remember that the flow goes along the arteries in the periarterial space. So astrocytes, which are support cells in the brain, are very important in maintaining that space. And when astrocytes become, they can change the acidic end feet can change their volume. So as the volume goes down, you have more volume in perivascular space. Potentially more fluid can also get in. So lots of things could alter that. And so what we found also published a couple of years ago, macrophages, which are immune cells, which are sitting in a perivascular space, are also controlling. So what they're doing is, it's very funny, right? So these are tiny, like chimney sweeps. They're sitting in the, so perivascular space is very busy. Lots of activities going on there. So cells produce their own local trash. How do you remove that? So macrophages are sitting there and actually eating all the extracellular matrix, which is being produced by the endothelial cells, the fibroblasts, the smooth muscle cells, whatnot. So if you remove those macrophages or you make them dysfunctional, you have accumulation of debris, if you wish, there. So this vessel becomes more stiff and can't really fully pump the fluid into the brain. So even so, such thing as a macrophage, which you wouldn't think that macrophages would affect flow, but they do.
Yeah. Now, when you say macrophage in the brain, you're really saying microglia.
No, 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 no. These are actually perivascular macrophages. They are
Okay.
sitting along the vessels, not in the brain, but along the vessels
Okay.
that are coming into the brain. Yeah, 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 yeah.
Good. Good.
These are the under-recognized, underappreciated heroes of the brain immune system.
Okay. Because these are the BAMs, the brain associated macrophages.
Well, yeah, you can call them BAMs. Yes, exactly. Yeah,
Okay.
yeah.
BAMs.
Even though I was a co-author on the first BAM paper, but I'm not crazy about the acronym BAM because BAM includes lots of different macrophages.
Okay.
So basically,
Okay.
BAM is basically BAM is everything in your head minus the microglia.
Are they like the bouncers? If you go to a bar,
Basically, well, they are well, you could call them that they're bouncers. Maybe bouncers are there are some other cells because there is actually entrance to the brain. Those are bouncers. Those mast cells are bouncers. Yes.
okay, all right. Now, you mentioned the astrocytes, and they have this star-like structure. They're critical. In fact, one of the papers we'll link to is this recent Nature perspective about how their role is much more important and outsized than previously estimated. They have at their tips, they connect with this important receptor that won a Nobel Prize years ago, the aquaporin 4,
Yeah.
right? And what does aquaporin 4 do and these astrocytes in
Oh, so it's a very good question. So aquaporin 4 is a channel, it's a water channel. So it moves water from one space to another space, and so it's in the brain. It is only expressed in astrocytes, and we know that if aquaporin 4 is missing or it's even mislocalized, then the water flow from extras from one compartment, which is along the vessels, to another compartment, which is the brain, will be affected. Will be destructed. So aquaporin 4 is one of the most important molecular mediators of this flow fluid from perivascular space to interstitial space.
And then some people have genome variants in the aquaporin 4 gene, which
Yeah.
sets them up for trouble, right? I mean, they're not common, I guess. These are relatively rare variants.
Right. Yes. Yeah.
Yeah. Okay. Now, um, if you're in atrial fibrillation, so your heart rhythm is not normal, there's been some recent papers to say, huh, that affects your lymphatic flow. Is that true?
Well, no, again, and a beautiful paper. If you're referring to Miken's paper in Cell with the norepinephrine signaling, it's fantastic story. I think it's definitely that data is definitely true. Yes, a very important study. I think. But again, no, we have such a so it's every time I try to explain the lymphatic to people, especially to skeptics, right? There's always a skepticism about one aspect or the other, and I say, no, it's such a complex story.
Yeah.
We cannot understand, even though it's been out there for now more than 10 years. I think 2012 was the first publication. We cannot grasp the whole complexity of it to the level of understanding every single aspect of it. So we're still in the midst of learning it and understanding what are the forces that are influencing. For example, I don't know if you saw our paper. So for the initial story was that when fluid gets into the brain, it has to go with what was the name of the flow? It was so it's not a diffusion; it's a convective flow.
Oh,
So it's,
right,
it's a
right.
flow that actually being pushed by something. But what pushes it wasn't really clear. So last year we published a paper where we said that neural activity,
Yes.
actually synchronized neural activity during sleep time, is maybe we will talk about this later.
The,
But that's
the,
actually one of
the
one of the
night shift.
forces.
That was the night shift paper. Yeah.
Right? Exactly. Yeah, exactly. Yeah.
And I do want to get into that because it's during sleep where all the action takes place. So, right,
Yes.
where you get that's when this whole clearance is occurring, and most of it is during deep sleep. That which
Yes.
usually in the earlier time of sleep doesn't
Yeah.
necessarily have to be, but usually, and it's the slow wave sleep. And why is it during slow wave deep sleep that this is when this the dishwashing takes place?
Yeah. Uh, Very good. So actually, funny. This morning, I was telling the story to seventh graders. I went to my son's classroom. The first time I did it ever, and to talk to this young kids, and I try to explain to them how important sleep is and what's happening during sleep time. So if you think, and I'm now just explaining it in the level of a seventh grader, so because it's just fresh in my mind, but so I apologize for listeners who are more advanced in their understanding. But basically, when we are active right now, lots of neurons are firing, and when neurons are firing, they produce waste, among which is amyloid. Right? Actually, David Holtzman's lab showed it very nicely that amyloid production is activity dependent. So during the daytime, we produce metabolites and all this. Now we know very well that the washing of the brain works more efficiently at night or when animals or humans are sleeping. I think there is no argument about it. Some people argue that it's only happening at night. I don't think. I think it's also happening during daytime, but it's way more efficient during nighttime. So what is happening at nighttime? What moves the flow from one place to within? So when we look at those cartoons about like drugs getting into the brain, we see this black void, two neurons, and drug is flowing. It's not really the case, right? So for those who don't know, if you look at the brain parenchyma, it is extremely dense tissue.
Yeah.
No free space to flow. But fluid somehow gets in and flows through. So what moves the flow? I had this exceptional postdoctoral fellow in my lab, who has now his own lab in University of Pennsylvania, UPenn. His name is Lifang Xiangqia. And he actually came to me and said, he goes, and he was coming from a sleep lab. And he said, listen, when we are sleeping, we generate, of course, waves. We all know that, right? So actually we can record your cortical activity. And based on the wavelength, we can say which phase of sleep you are in. So when we are in REM, you have faster waves. When we are in a non-REM sleep, in deep sleep, we have slower waves. And he said that these slow waves, they generate a lot of force because it's a synchronized activity of neurons that are firing all together. And the firing means you release a bunch of ions into the cellular matrix, which are flowing. And ions are charged molecules, and water is a polar molecule. So water will most likely follow those ions. So when measuring voltage in the tissue, we're measuring a flow of ions. So could it be that these very strong waves that's what's carrying the waste or the water through the brain tissue? And I thought it was a great idea. Let's try it. And so he actually showed it very convincingly in his paper that came out last year in 2024. He showed that when neurons are synchronizingly firing, that force generates force to move the fluid through the brain tissue. And if you silence those neurons, then you have less water goes through the tissue. So when we are sleeping, when we're going into this deep sleep, and we generate this very high potency, slow waves, we believe this is not the force, but this is one of the
Sure.
forces that moves the flow through the brain. That's
Yeah.
why it's so important to get good night's sleep, with all the right amount of deep sleep.
Right. And when we're awake, we've got higher levels of norepinephrine and other both physical and many different changes that would not be that kind of magic of deep sleep that you're referring to with the
Right.
kind of synchronized force of neurons. Now, um, we should all be getting as much deep sleep as possible because of this, right?
I would believe so. Yes, unless you are a natural short sleeper, and
Oh,
then
well, that's
and
very,
then
yeah, that's very rare. These people that can function on 4 hours have
yes.
perfectly intact, and maybe they have accelerated clearance of their.
So exactly. So what we are, so actually there was beautiful work. It's a lab at UCSF. Actually, they crossed this natural short sleeper mice with Alzheimer's mice.
Mm,
And the resulting mouse actually was developing less pathology. So it's kind of out. It's a bit of counterintuitively because mouse sleeps less, yet has less pathology. But if you think what you just said, if you think of a sleep not as a sheer number, but efficiency, how efficient you are during your sleep to cleanse your brain, if that's one of the reasons why we sleep,
yeah.
like if you're efficient, if you can do the job in 3 hours, then why sleep for 6 hours, right? But
Right.
but you're right, most likely you can't, and so you need enough sleep there to clean your brain more efficiently. But yeah, my dream is to be able to develop something that will clean our brains in 1 hour, and so we can sleep 1 hour and then go back to life.
Well, I when I was doing the research for the book Super Agers, this is where it came alive about how important deep sleep is and how we both have used the Aura Ring and other ways to measure
Yes,
that. And I mean, learning about your deep sleep is a practical import to try to get it. If it's good, if it's a good number percent of your sleep, that's great. But if it's low, like mine was, and working on ways to get it maximal is really helpful. And for me, the biggest thing, as we discussed before we got started, was just getting on a very regular schedule. Made a huge
yeah.
difference in getting into that deep sleep. I just think about it every time now, Jonathan, because of your work and others, that I'm getting a good cleaning of this
Oh,
junk
for
that I don't
sure, yeah.
want. Because
But you know what?
people don't realize that this is pro-inflammatory in the brain. If you don't
Oh,
get that
for sure,
stuff
of
out,
course.
it's the worst thing you could have for your
It's the
brain.
worst thing. Yes, yeah, yeah, exactly. Yeah.
And one night of bad sleep. Can you comment about one night of bad
Well,
sleep?
no, okay, let me, I'm not a sleep researcher, right? So I mean,
Right.
sleep, we're looking into sleep, of course, but I'm not a sleep researcher. So I think that maybe I should not be making those comments also because there are so many nights that I don't sleep well at all. So I hope that these people are wrong saying one night of bad sleep is terrible. But I can tell you, we have this very exciting new work, which we haven't even submitted yet, but I can just maybe tell you, reveal the secret because I think it's so exciting. So you know how old people, they need more naps during daytime because they don't sleep very well at night. So there's more pressure to sleep, right? So we also don't understand what is pressure to sleep. What is the pressure? So we think that pressure is actually dirt in the brain that needs to be cleared. That's the
Mhm,
pressure potentially.
mhm,
It's a good
mhm.
hypothesis. So
Interesting.
when you look in old mice, they have truncated sleep during their sleep time, which is our daytime, and then they take more naps during their activity time, which is our nighttime. So they more naps and less well sleep during. So if we take these mice and we just improve their brainwashing through improving of lymphatic function, we normalize they're no longer taking naps during daytime.
Interesting. Interesting.
And
Wow.
vice versa, if you take young mice and you destroy their lymphatic vessels, then you start seeing more napping during wakefulness time,
So there's somehow a signal from not having incomplete waste removal that it suggests that you need to get a nap during the day even.
which we not fully understand what exactly the signaling is. But yes,
Yeah,
and
yeah.
as you know, during naps you don't really get into deep sleep. So even though you are resting, but you are not really probably cleaning it very efficiently.
Yeah, yeah, depending on how good your nap is, how
How
long it.
long? Yeah,
Yeah, yeah.
exactly.
I've seen some of my naps were magical for catching up on some deep sleep, surprisingly. But yeah, you're right.
Well, people say that Einstein used to do micro naps, right, of a
Oh,
couple of seconds. I don't know how you do
I
it,
know
but
that would be hard to get a lot of clearance from that. Now, it's fascinating. I want to get into now this border zone and the efflux. So the lymphatics have done their thing and all these things that we've been talking about, and now we're getting the stuff out of the brain.
right.
And as you pointed out, there's the skull itself, the bone marrow of the skull, the meninges, the dura. This is an immune reservoir.
Yes,
This is our immune cells are hanging out, and they're getting a moment to moment intelligence report.
absolutely.
Stuff is coming out, right?
Yes, I love the analogy. Yes.
So you're basically getting this incredible surveillance, And here's your immune system. It's ready to act. And you've got these regulatory Treg cells. You've got a rich system here in the brain border zone to
Yes,
to protect the brain, to
right.
go into the brain
If
to
you need them, if you need them
if
to,
we
yes.
need it, right?
Yeah,
Because otherwise, it's coming down the meningeal lymphatics and getting out to the body, right, through
right.
the
Yeah.
lymphatic system. So, what about that surveillance? What is the main things we should know about that border zone surveillance process?
Yeah, so like you said, right, so so basically in normal tissues, immune cells can get inside the tissue, but in the brain, then surveil and understand what's happening. Is there any abnormal proteins are being presented, or is there something is wrong? But in the brain, because we don't have access of immune cells to the brain tissue normally in healthy condition, or it's very restricted, then what you do is you take this immune signature, you take it out into the borders, and that's where you present, and that's where you have what we call it immune barcode. And we believe that, and I think everybody would agree that every tissue, of course, will have its normal immune barcode. Now, the moment tissue is getting into distress, whether it's disease is starting, or could be injury, could be tumor growing, could be infection, whatnot, your what you are presenting is completely changing, because the way to what is being presented has to be processed, and processing is affected by infection, by temperature, by lots of different things. So the moment there is a disease, you start seeing change in this immune repertoire of what you are being presented. So basically, the immune system probably detects the problem way before your physician will detect your problem.
Right, right,
So the question is, how do we get in and understand what is being detected there, but also can we actually restore that signature back to normal? So if you think about it, if you think of a barcode, think of a disease with a barcode where some of the black bars are erased and you can no longer read it with a normal scanner. So what do we do? Can we replace those missing proteins to reinstate the barcode and reinstate the normal immune surveillance of the tissue?
Yeah, no, this is also that brilliant paper you had in immunology in May, where you
Yep.
talked, it was the immune code was the key concept. And what I love in that, we'll put in the link, you have this figure where you show barcodes, theoretic barcode for healthy, for autoimmune disease, let's say multiple sclerosis,
Yeah. Yeah.
or aging. Tell us the differences of how the barcode in those different states would lead to the whole clearance and effects on the brain and the outflow.
So I'll give you one example with one protein that we actually published with. So there is a one peptide, one tiny peptide, which coming from our myelinating glia cells. And that peptide actually does something very important. It makes sure that the autoreactive cells around the brain are all turned into like regulatory suppressive phenotype. So basically, it neutralizes all the autoreactive T cells into more suppressive, so they cannot damage the brain. Now, what's happening in Alzheimer's mice, but also in aging mice, and we also see it in mice with multiple sclerosis, that protein disappears completely. That protein disappears completely. It's no longer there. So your guardian peptide, which we call it guardian because it was there to protect the brain from
Right.
immune attack, is now disappearing. So now your immune response around the brain is now more pro-inflammatory. And it's just one example, but there are many of those guardian peptides, and they are all changing in their presentation. So this signature, the immune signature outside the brain, we believe is very important to make sure that your immune system is in check. Because remember, the immune system is a friend. It's not the enemy, but
Yeah.
its goal is to respond to a problem. And when immune system responds to a problem, there is collateral damage. So now, if the problem is chronic and is constantly there, then the collateral damage will also be constant. So
Right.
while you need your immune system, it needs to be in check and not just go on uncontrolled. I like our analogy when we published a paper when we made an analogy of immune system to construction workers. So when you wake up one day and you see construction workers on the street, on your street, you will not be happy because it will be noise, traffic congestion, right, dust and whatnot. But they came there not to make your life miserable. They came there to fix the very important problem.
Right.
And same with immune cells in the brain. So when they go there for a good reason. But you also want them to be out as soon as possible. You want to make them very efficient and get them out. If they persist there for a very long time, then the problem, of course, overwhelms the brain, and then it just then by themselves become a problem.
In that important figure where you show the difference between an autoimmune disease like MS and aging, they have similar. I mean, they have different barcodes, of course, immune barcode. They have an antigen presentation, of course.
Yeah.
But the difference that you show there is that the autoimmune has intact outflow of meningeal lymphatics, whereas the aging has decreased lymphatics. Is that Is that the right representation?
It is very right. It is maybe even more than that. It is possible that in autoimmunity we may be having over functioning lymphatics. And so there is a too much proteins are being we're being the brain is being too generous with the immune system and giving them maybe a little bit too much of its content, triggering more activation of the immune system. But with the neurodegeneration, yes, what we see, and I think it's now repeated by so many different labs, we see a dysfunction. But every organ, every system deteriorates with age. Unfortunately, I mean that's just a fact. Some of us are super agers, most of us are not, right? So,
Right.
like I told today to the kids, I said, if we live long enough, we'll eventually develop dementia and some sort of dementia. We'll eventually develop it. So unless something unique comes and we change it completely, but right now that's the if you live long enough, you will develop one of those diseases. So one of the aspects that goes down with aging is of course the function of lymphatic vessels, and that, if you wish, is your drain, right? So if you think, I know you bring an analogy of a dishwasher, I like it, but I prefer an analogy of a sink. So you have a faucet that gives you clean water. That's the production of cerebrospinal fluid. That's our choroid plexus. And you have a sink, which is our brain, and then you have the drain, which is the
Okay.
lymphatic vessels,
Okay.
right? And so if your drain is clogged and you're washing your dishes in that sink, all the dirty water will remain within the sink. You can play with the faucet; it won't help because the drain is not functioning. So you need to go for the drain. You can, of course, take a little bucket and take the water out of the sink, but it's not gonna fix the problem. The problem is with the drain, and that's where I think lymphatic system is coming as a very important handle, not just to fix the drain, but also to fix the flow through the brain. So
Oh, sure.
lymphatic and glymphatic are linked. It's a one, like one is the beginning, one is the end, but it's a one big system. And I don't know if you heard about this Chinese, I call it Chinese surgery, but it's surgery that's done in China primarily, where what they're doing is they're taking the lymphatic vessels which are coming out of the brain and they connecting them into the blood vessels to make the flow faster. And you people can search it online. It's not as it only has been done in China. In several hospitals now, there are a couple of publications, but a lot of data online. They're calling this LVA for lymphatic vascular anastomosis, LVA-AD for Alzheimer's disease, LVA-AD. If you Google it, you'll find much information. It is very, very promising. I'm being very cautiously optimistic. I want to believe that this is really as amazing as described. I want to understand better how long it will last, how efficient it really is. So we're doing something similar in mice, and the results are very, very promising. Very, very promising.
I mean, this is really striking
It
because
is. It is.
the
Yeah.
idea now that we finally have learned about, as you say, the sink and the drain and the faucet, but that we could fix the drain with
Yeah.
the lymphatic venous anastomosis and get the superflow clearance, and then ablate or even treat Alzheimer's.
That's what they're claiming. Yes, that's what they're claiming. And again, the results that they're showing, and many of them are now published in peer-reviewed journals. The results are very, very interesting. There, I cannot reveal too much information, but there should start clinical trial. Actually, I think it was already published. There was a Science magazine a couple of weeks ago. They discussed there's a clinical trial which will start hopefully at Yale University very soon.
Okay. Actually,
So with
they're gonna take Alzheimer's patients. They're going to do this surgery and see if they respond. But don't you think that, Jonathan, that when you have so much inflammation in the brain and all these misfolded proteins, it's too late to try to reverse that?
I agree with you completely. That was my major concern. I couldn't understand how come they showing such amazing results. So what will happen if I have to predict the future, and nobody, and there's no prophets, of course, among us, but what I think will happen is when you open the drain and then all the dirty water comes out, it doesn't make the sink clean, but at least it removes the dirty water. Now you need to still clean the sink, right? You need to bring the soap and the water and all this to clean the actual dirt around it. So you need to restore lymphatic function around the brain, not just to kind of, you need to, like you said, you need to control inflammation. But I think when you restore lymphatic function, you will also subsequently restore inflammatory response.
Okay. Well, this is
So it probably will be a long process, but I don't think that the surgery by itself will be sufficient. I think
yeah.
that there is more to it that needs to be. But what I was thinking, all the anti-amyloid therapies that we are using now, they are efficient but not perfect. But what I'm thinking, if you restore the flow through the brain and you provide those drugs, will you augment their function? I think that you will.
Well, what you're bringing up is really interesting because the efficacy of these monoclonal antibodies to amyloid, there are two that are approved, as you know, they can take out all the amyloid,
So
but the cognitive doesn't. It's very minor.
yes,
It's a
yes,
very complex treatment. It has lots of risks for hemorrhage in the
yes.
brain and whatnot. But that's what's interesting is you get the amyloid out and you don't get the clinical improvement. And this,
But you don't. You dissolve plaques. You don't necessarily get amyloid out. You dissolve the plaques, right? So
okay,
the monomers are still circulating in the brain, which are very toxic.
Right.
So I think, and again, I'm not a medical doctor, right? It's my understanding as a scientist that I think
Sure.
if you improve the flow, if you really now let things flow out of the brain while the plaques are also dissolved, I think it will probably combinatorially work very, very well. It will it
Okay.
will have
I'll,
synergistic effect.
I'll buy that point you're making, which is that combination. Um, but this, don't you think it would be better to in people who are very high risk for Alzheimer's but are still cognitively unimpaired? Is that would be the group that would might benefit the most?
From the lymphatic surgery, I mean?
Yeah. Yeah.
It is possible, So when you are young, the system is functioning. So, if you take Alzheimer's mice, and in mice, of course, we engineer them so that they develop Alzheimer's plaques very, very early. If we just work on enhancing lymphatic vessels in young mice, we don't really see benefits. We need to wait until they are very old, and then we can see some benefits. But if we combine them with anti-amyloid therapy, yes, with lymphatic being more efficient, anti-amyloid antibodies are working more efficiently as well.
Now, I'm just thinking about this surgery, Jonathan, and it sounds pretty delicate. You're basically taking um, connections to the from the uh, efflux of the brain into the veins. I mean, this doesn't sound like a simple operation at all.
So that's what I thought. No, so in mice we are doing it slightly different. We're calling it lymphatic bypass, but it's very similar in the idea. So if you think about it, the lymph node by itself is actually provides resistance. So we're doing very similar surgery, bottom line. And in mice it works really, really well. Very, very promising results. Very promising
Mm,
results.
mm, mm.
So in humans, so Eric, I'm not worried. I mean, we're working at Yale, working with some of the absolutely best surgeons in the world. And so but whether, every human being is different. And when you open, I think it will, in mice, all the mice are the same. We work with identical twin mice, right? As you know, but with humans, they're all different. So there will be some variation.
Yeah, yeah.
I'm wondering how can we really make this, with this therapy, reproducible between person to person? That I
Right.
do not know. But
Well, this is an exciting potential you're bringing up.
exactly, yeah, that's
We
why
can,
that's why the clinical trial hopefully will start and they will see how reproducible this data could be.
yeah, I mean, I have to say it has a lot more appeal than you remember when they were doing stents in the carotid arteries for multiple sclerosis started in
That was a scam, right? As far as I know, but
this one has a little more to it. Okay, we'll see how it plays out. Now, the opposite of that, as it turns out, if you take Ambien, the studies show that you basically stop the drainage. You heard the person feels like they had to sleep, but their drainage is poor. Is that true?
well, again, I don't want to discuss other people's words that I'm not involved actively, but I mean the data looks interesting, and again, we know it very well from Mike and Idegard's work, from our work, from many other labs. You can put a mouse on anesthetics, and depends which compound you use to anesthetize the mouse. Some of them will more mimic natural sleep,
Yeah.
and you will have beautiful synchronization of neurons, beautiful waves, and you will have very good flow through the brain. Others will stop the system completely.
Right.
Right,
Right.
so there are different anesthetics that you use that there is no flow at all. The
Well,
whole thing just stops.
no, you had a section in the neuron paper of the
Yes, where we discuss
anesthetic.
differently. Exactly.
Yeah,
Yeah.
yeah, and I think, the same thing is the case with these drugs that are and supplements that are being advocated. People need to be wary of that because they can have very, untoward effects or heterogeneous effects, as you say, so very
Yeah.
important.
What I tell myself, right? By me sleeping 8 hours at night doesn't mean that I get enough of a good sleep, of a
Oh,
deep
yeah,
sleep to
yeah.
clean the brain. So, I used to do something which at night, I would like to have like a little bit, a cup of glass of whiskey. I mean, a little bit of whiskey, just to kind of help me go to bed. But it doesn't do good to your sleep patterns, right?
No,
So
no.
actually, it kills your deep sleep. So, that's just one example where you think you're helping yourself, but you're really not.
You're bringing up the other thing, which is more important than the Ambien, which is alcohol does decrease deep sleep, no question. And
Exactly.
people,
Yeah.
if they start tracking it, they will see that. I mean, I certainly learned about that too. And not if you're trying to maximize your deep sleep, that's the last thing you want to do.
Exactly. Yeah. Yeah.
Obviously,
But again,
you want to enjoy life, but there's another price to pay, and deep sleep is part of it. Now, this has been a fascinating discussion because it's leading us to new strategies. I mean, who would have thought that your discovery a decade ago of meningeal lymphatics would ultimately lead to an operation to improve
so,
clearance?
Eric, I should give credit here if you don't mind. Just very, very quick story. So I was in University of Virginia, which I know is also dear to your heart. And when we just found the vessels, my chair back then, who unfortunately passed away a few months ago, Kevin Lee, said, we must show it to our chair of neurosurgery. So neurosurgery at UVA had this very famous chair, John James Sr. He was like,
Oh, yeah, yeah, yeah.
exceptionally famous guy. A bunch of chairs in the US of neurosurgeries were trained with him. So we came to him. He was already quite sick back then. He was with glioblastoma, but he was still functioning and a very smart guy. So I showed him the story. And I am a neuroimmunologist, so I'm thinking lymphatics, inflammation, MS. And he looks at this and says, "Oh, it probably will be important for Alzheimer's." I'm like, "What is he talking about?" And he said, "I actually thought that Alzheimer's will eventually will be a neurosurgical disease."
Wow.
And
Wow.
I left that room and I said, "What was he talking about?" And then, today, unfortunately, he passed away. But I would love to know if he was alive. I would love to go and say he literally was way brilliant. He could see he saw the future coming. Yeah,
It's wild. Totally wild.
he was wild. Yeah.
Before we wrap up, I want to just zoom in a little bit on these cells with you. So we talked about the brain-associated macrophages, how important they are. We talked about astrocytes and how critical they are. I just want to talk about the microglia. The microglia are a big part of the brain cells, if you will. Their function, and then you also have published recently, and others are interested in this field of microglial transfer replacement. Tell us about this microglia story. There's a lot going on there too. It's different than what we've been talking about, but just to round out some of your work.
It's different, but not really, right? Because microglia this is the only major immune cell that resides in the brain. So these are the residents of the brain, and immune cells. What are they doing? They are cleaning the environment. So I think they are also cleaners, and we know that, for example, to dissolve the amyloid plaque, microglia probably are involved in all this. And if you activate microglia properly, they will do a job very well. If you activate them improperly or in some crazy way, they will be super too active or too inflammatory. Of course, they can also damage the brain. Microglia can eat synapses, for example, which are the interaction between neurons. Could be a good thing if it's a bad synapse, could be a bad thing if it was a good synapse, right? Microglia can produce different, we published, could produce different molecular mediators, which will then talk to neurons directly and change function of neurons. So these are immune cells, which are very, very important in the brain. And actually, the funny story is that we used to call them resting microglia. Means that microglia we used to do like slices of the brain, and we call them resting microglia. And then 2005, the group of Wayne Biao-Gan at NYU, the first author was Dimitrios Davalos. Actually, they published a paper where for the first time they looked at microglia in a living mouse, and what they saw is microglia were constantly moving those little processes, and they were anything but resting. So constantly surveying the environment, and I believe that this is a if we're speaking about clearing the brain, this is the micro clearance, and then what we spoke about is a macro, right? So this
Yeah.
is kind of the movement flow, and then microglia are the small are kind of more cleaning the janitor that comes into your building to clean more local environment, whereas the flow is something that cleans the streets and whatnot of the town. So yeah, I still
So
think that they are. And the beauty of microglia, like you mentioned, is that you can replace them because they are immune cells of the brain, and you can remove them and then you can bring new ones that are more efficient. And I mean, there's now lots of also very interesting stories coming out. Some of them are from WashU. I think big paper is about to come out hopefully soon, where you can actually engineer them. Like we know CAR T cells, but you can
yes.
also maybe CAR microglia. And so to make them more efficient in particular task.
So the story here would be you would get these cells, whether they're engineered microglia, abundant in the brain, or increase the functionality of those already in the brain, and that would help get another way to improve the clearance.
Yeah.
Different than the surgery that you just were describing earlier, but
Yes. Yeah.
basically an in the brain strategy rather than from the efflux of the brain. I think it's very intriguing. It's more of a cellular approach rather than a surgical approach.
Yes. I mean, again, because every neurological disease is so complex.
Yeah.
I think that at the end of the day, if we really want to, first of all, I believe that we probably will be more efficient in preventing or delaying the onset of disease than curing the ongoing disease. But I think if we want to cure ongoing disease, we probably will need to use a bunch of different approaches eventually.
Yeah.
So it may be replacement plus the drainage plus the anti-amyloid antibodies plus whatnot, and to kind of target the disease from many different angles.
Now, we've talked a lot about trying to prevent cognitive impairment, Alzheimer's, and better clearance. I just want to go back to the autoimmune story because we haven't really. How do we fix this problem, knowing that what you know about the immune code and the brain immune interactions? How can we? I mean, this is another dreaded disease, of course, MS. How can we make a dent in that?
Yeah, not only MS, there are so many other diseases which
Oh
are
yeah,
based on
yeah,
abnormal inflammation around the brain. And so that's something that's very dear to my heart because, like I said, I'm a neuroimmunologist. I mean, this is what the main focus of ours. We even have company now that we're trying to push into like real therapy and medicine. And that's what excites me tremendously. And interferes with my sleep. So when I go to bed, I think about this. So again, one thing that we found is that we found those molecules which we call guardian peptides, which are really balancing the immune response around the brain. And we showed in animal models of multiple sclerosis, if we provide more of this, so what's happening when the cells are under immune attack, if you wish, or there's inflammatory condition, or maybe even infection, like EBV infection, for example, the presentation of the molecules that you see changing completely because cells are in different state and they are not presenting. So basically, if you think about it, when you have an attack, viral attack or bacterial attack or whatnot, you remove all the breaks. And along those breaks, you also remove the peptides, which are controlling balancing the immune system.
Right.
So what's happening is that our guardian peptides are disappearing completely. And we show that if you now provide these guardian peptides back into the brain borders, then they rebalance the immune system, the immune response,
So,
and suppress ongoing autoimmunity.
so basically, you're trying to provide that constant surveillance, the intelligence report that things are not really off in the brain. You're giving guardian. How would you conceive of that as a treatment? How would you
So
do it?
So we would just so again, what we're doing it in mice, we're doing it two ways. One way maybe is a bit more straightforward. We're delivering it in particular particles into the cerebrospinal fluid.
Okay, so it has to be intrathecal. Yeah, yeah,
Yes, and then through CSF it will get into the brain borders, and there will be uptaken as if it came from the brain. We're tricking the system, like you said,
sure.
and then being presented, and then it suppresses it actually works very, very well. The other way is to give it as a vaccine.
Okay,
So you increase the number of these cells in the periphery. You will still probably, I think that the best way for humans probably will be vaccines. So you have the reservoir of these cells, or you increase the number of these cells that are responding. But then you probably will need also to provide the protein, the peptide, whether into the CSF or peripherally. Again, in humans, it's probably way more complicated. But there's ways you can maybe do nasal delivery. You can maybe do we do now some crazy things. We do actually transcranial delivery because if you think about it, right? If you put stuff, skull is a porous bone.
yeah,
So in theory, you can deliver molecules across skull.
right.
And I once gave it to I was giving a lecture to NASA, and so then they wrote article, "A shampoo for brain disorders." Would be amazing.
It'd be pretty tough shampoo, but you're bringing up a really important point, Jonathan, and that is that we don't take advantage of the skull meninges way to get in to the brain. The other thing that
Well, we do now in the lab. We're looking into
oh
it.
yeah,
We're
in the
getting into
lab,
it.
but we haven't gotten there yet
We have not. No.
yeah, but what I like about the vaccine, I just wrote a piece in Ground Truth about the tolerogenic vaccines. We're trying to get tolerant. When you know the antigen, as we do know with Epstein-Barr and multiple sclerosis as one example, we should be able to come up with a tolerogenic approach. To
Yeah.
basically, what you're saying is that you don't get this immune response. You basically don't recognize it as an alien.
Well, so
Not
no, so here it's even more because what's happening here, these are self proteins which are being generated spontaneously and naturally.
yeah.
And the only immune system they drive the suppressor cells. So they will not drive bad autoimmunity, they will drive the suppressor cells. And so we have the peptide, it's not artificial one, we did not come up with it. It's endogenous. We all have it in normal, healthy state.
So,
And what's
yeah,
happening
but
with disease, we're losing it. So we're just giving it back to you.
no, I was just thinking you could do both.
Oh,
Yeah,
for sure. Yes, of
yeah,
course.
yeah, yeah. But no, I mean, you're bringing up some pretty exciting, optimistic outlook for what we can do for preventing Alzheimer's and coming up with much better preventions or treatments for autoimmune diseases that involve the central nervous system. So, here's a question: could a recombinant polyclonal immunoglobulin work? Uh, I don't know. But yeah, we like questions. That's fine. But we're just about to wrap up. This has been terrific. We've had like 550 people join us. They've been commenting about how much they've enjoyed the interaction, which obviously we appreciate. Jonathan is as good as it gets in this field, and so glad to have this conversation and learn some things that are on the horizon out there. Potentially even getting into clinical trials soon. And the metaphors here, there's no shortage of metaphors. I guess the one that sticks with me better than my dishwasher at night is the faucet, sink, and the drain.
Yeah, I also think
Well,
I like the sink as well. Yeah, yeah, yeah. It's
yeah,
more, especially when I show it, sink with dirty water in it. We all live, we all experience it. It's at some point, right?
yeah, yeah,
Yeah.
really. Did I miss anything before we close, Jonathan?
No, I think we covered it all. It was pleasure to first of all to meet you in even though it's virtual, but to meet you like this. And so, thanks for highlighting our work. Thanks for inviting me. It was a pleasure to discuss the science with you.
And last question from the questions. There was a recent paper, as you know, in Nature about lithium orotate. Any comments about that?
I would rather not comment on that. I would
Okay.
rather Alzheimer's expert comment on that. It's an
Okay.
interesting story. I read it, but I cannot
I
again. And
yeah,
it's not my field and my science. It's hard to comment.
right. No, I only ask you because I want to show that we do respond to questions that come up.
Oh yeah, for sure. Yeah. Very interesting
That's all.
story. But we need a real Alzheimer's expert to discuss that.
Right,
That
right.
yeah.
Absolutely. Jonathan, thank you so much. As I say, we'll post this with links to these incredible papers you've had recently. Keep up the great work. You've got phenomenal momentum, you and your team there, and we'll look forward to all these advances you've been talking about. Thank you.
Yeah. Thank you very
Thanks
much. Thank
everybody
you. Thanks very
for
much.
joining us today. Really appreciate your support of ground truth.
Thank you.
Bye bye.
Thanks. Bye bye.